Assalamualaikum and hi everyone. We are from Group 2 and our chosen topic is proteomics and interactome. Today we will be presenting our analysis of the research article titled Quantitative Interactome Proteomics Identifies a Proteostasis Networks for GABA A receptors. This study focused on understanding the proteostasis mechanisms of GABA A receptors and their implications in neurological disorders like epilepsy. Our group members consist of myself, Nur Hadira, one Nadatul Alpha, Nur Hamiza, Nur Afza, and Nur Izati. I will begin with the introduction and objective of the research. Firstly, what is GABA-A receptors? GABA-A receptors are the critical neurotransmitter gated ion channels in the mammalian central nervous system. They regulate the balance between excitatory and inhibitory signals, which is essential for proper neural function. The disruptions in these receptors is linked to neurological disorders such as epilepsy. Despite the importance, the processes governing the biogenesis of GABA A receptors, specifically folding, assembly, trafficking, and degradation, are not well understood. This gap in knowledge is particularly significant for misfolded receptor variants, which contribute to disease pathology. This research uses a proteomics-based approach to systematically map the proteins that interact with GABA-A receptors, also known as the proteostasis network. It focused on both the wild-type receptors and a misfolding-prone mutant, which is A322D, to identify the key difference and their potential implications. By understanding the interactions, the significance of the study is to provide insights into the molecular mechanisms underlying GABA A receptors proteostasis and highlight the potential therapeutic strategies to improve receptor function and address related neurological disorders such as epilepsy. The objective of the study are firstly to identify and characterize the network of proteins involved in the folding, assembly, trafficking, and degradation of GABA A receptors using a proteomics-based approach. Second is to compare the interacting proteins, which is known as interactomes, of wild-type GABA A receptors and a misfolding promutant A322D. And lastly is to understand how the proteostasis network regulates GABA A receptors, biogenesis, and function. Below here, we've also attached the diagram showing GABA-A receptors proteostasis network. The diagram illustrates the pathway regulating the folding, assembly, trafficking, and degradation of GABA-A receptors within the cell, emphasizing their biogenesis in the endoplasmic reticulum. Firstly, the individual subunits of GABA-A receptors fold in the endoplasmic reticulum. The properly folded subunits will assemble into a heteropentamer in the ER for subsequent trafficking to the plasma membrane. But the unassembled and misfolded subunits will be subjected to the ER-associated degradation pathway by the proteasome or the lysosome-related degradation. That is all for the introduction. Next, my teammate will proceed with the methodology and results of the study.
Hi everyone, now I will be presenting findings from research on the proteostasis network of gamma aminobutyric acid type alpha 1 receptors focusing on both wild type and mutant forms. Let's walk through the methodology and results using the figure on the slide. Starting with the methodology, HEK293 T cells were transfected with either wild type or mutant alpha 1 receptor constructs. Stable isotope labeling by amino acid siRNA-C labeling was applied to differentiate samples by incorporating light and heavy isotopes into proteins. The samples were then subjected to immunoprecipitation, as shown in this figure B, to isolate interacting proteins. Mass spectrometry, as shown here, was used to identify and quantify this protein. Finally, bioinformatic tools were employed to assemble a detailed protein interaction network. Okay, now let's examine the results. For the figure B, the interactome analysis identified 1 to 5 interactors for the wild-type alpha 1 receptor and 105 for the A322D mutant with 54 shared interactors. Chaperones like HSP70 and HSP90, degradation regulators such as DRIM2-1 and SEL1L, and trafficking factors like SEC16A were identified as central to the proteostasis network. The RIM21 in particular played a crucial role in selectively degrading misfolded alpha-1 subunits, showcasing its importance in maintaining receptor homeostasis. From the bar graph figure E and F, you can see the breakdown of interactors across cellular compartment, including the proteasome, mitochondria, and endoplasmic reticulum, emphasizing their role in protein folding and trafficking. The bioinformatics analysis also highlights processes like proteolysis, protein folding, and transport. In conclusion, this research established a detailed proteostasis network for gamma aminobutyric acid receptors, revealing mechanisms regulating folding, trafficking, and degradation. Importantly, it provides insight into targeting misfolded proteins for therapeutic intervention. Now I will pause to Alpha to talk about principles and its application.
Thank you, Elsa. Next, I will explain the principles of quantitative interactome proteomics and its applications. The study employs quantitative interactome proteomics, specifically immunoprecipitation, tandem mass spectrometry, IPMS or MS, combined with stable isotope labeling by amino acids in cell culture, SciLab. This method identifies proteins interacting with GABA A receptors, aiming to map their proteostasis network, which regulates folding, trafficking, and degradation. SciLab enables precise comparisons of proteins interactions in cells, expressing either wild-type or mutant receptor variants by differentiating them with isotopic labels. Furthermore, the principles underlying this method include protein-protein interaction identification. By immunoprecipitating GABA A receptors, associated proteins are isolated and identified via mass spectrometry. Next is quantitative analysis. SciLab provides a comparative and quantitative approach to identify and measure interaction differences between normal and misfolding prone receptor variants. Last one is proteostasis network mapping. Bioinformatics integrates data to model molecular pathways involved in receptor homeostasis, such as folding, degradation, and trafficking mechanisms. Next, we will look at the applications. This research has significant applications, especially in the medical and neurological fields. First is neurological disease treatment. For instance, insights into the proteostasis network of GABA A receptors may lead to novel therapies for genetic epilepsies and other disorders involving these receptors. By selectively targeting specific interactors like TRIM21, researchers can restore functionality to misfolding prone receptor variants without affecting normal proteins. Another application includes drug development. The study provides a foundation for creating small molecule drugs to fine-tune proteostasis networks. Moreover, these drugs could enhance folding capacity or prevent degradation of pathogenic receptor variants. Last one is for broader implications. Understanding receptor biogenesis and misfolding mechanisms can impact treatment strategies for other protein misfolding related diseases such as Alzheimer's or cystic fibrosis. Now, I will pass to Hamizah.
Okay, thank you Alpha for the explanation. Next, I will highlight the strength of this study and why they are significant. One of the most notable strengths is the use of scilab-based proteomics. This advanced method enabled the researchers to identify 176 proteins that interact with GABA A receptors. It's precise and highly effective, allowing a detailed comparison between wild-type receptors and mutant receptors, like the misfolding prone A322D variant. Another advantage is that the study highlighted specific therapeutic targets such as TRIM21, E3-ubiquitin ligase. By targeting TRIM21, it may be possible to rescue misfolded receptors and restore their function, which could be highly beneficial for treating conditions like epilepsy. Finally, while HEK293 T cells are not new neural cells, the researchers demonstrated that their protein's expression level closely match those of human brain tissue. This support the translational relevance of the findings, making them applicable to real-world neurological cells. While there are strong points, no study is without its limitations. For the disadvantages of the study, the main limitation is the use of HEK293 cells. Although these cells are great for control experiment, they lack the complexity of a native neuronal environment. This means the study couldn't capture key synaptic interactions, which are crucial for understanding GABA A receptor function. Another disadvantage is that variability across proteomic studies can arise from differences in purification methods or cell models. While this study provides strong insight, its findings might not be aligned perfectly with the previous research, especially those using neuronal models. Now, let's explore how this study fits into the larger body of research. On the supporting side, this study confirms the critical role of proteostasis mechanism in regulating protein folding and degradation. For example, molecular chaperones like HSP70 and the ERED pathway were identified as key components in maintaining protein quality control. These findings align with earlier research, reinforcing the importance of proteostasis in neurological disease. However, there are conflicting points as well. Compared to studies that use neuronal models, this study identified fewer synaptic proteins as interactors of GABA A receptors. This discrepancy is likely due to the use of HEK293 cells, which lack the native neuronal environment. Despite this, the study's findings remain valuable because of its focus on proteostasis, which is consistent across cell types. Now, let's discuss the limitation of the research method. The key limitation is the use of HEK293 cells. While these cells allow for precise control over the environment, they don't fully replicate the brain native environment. This means important synaptic interactions that occur in neuron may have been missed. That said, this choice is justified because HEK293 cells do not naturally express GABA A receptors. This allowed the researchers to introduce specific receptor variants without interference from endogenous proteins. Additionally, the similarity in protein expression between HEK293 cells and human brain tissue adds relevance to the findings. In future studies, researchers could use stem cell-derived neurons, which would be better replicate the brain environment. This approach could provide more comprehensive insight into both proteostasis and synaptic interactions. So that's
Next, I will explain databases and bioinformatic tools that is used in this article. The first one is David. David is a database for annotation, visualization, and integrated discovery. It is facilitate functional enrichment analysis of large datasets, helping to interpret gene and protein lists. The second one is Proteomics DB. Proteomics DB provide MS-based data for protein expression and abundance across tissue and cell lines. It is offering a detailed view of proteomic landscapes. UniProt also used in this article. UniProt is a created database that provides comprehensive protein sequence and functional annotation data, serving as a central resource for protein research. GeneCard is an integrative database of human gene, offering genomic, proteomic, transcriptomic, and functional information in accessible format. The last one is Protein Atlas. The Protein Atlas maps the human proteome using transcriptomics and proteomic data, providing insight into protein localization and expression patterns.